Welcome to this lecture on bioinformatics applications and limitations. Imagine you are handed a massive, disorganized library containing billions of books, but none of them have titles or authors listed on the spine. If you had to find a specific recipe for a cure hidden in one of those books by reading them one by one, it would take thousands of lifetimes. This is exactly what the human genome looks like to biologists. Today, we will explore how bioinformatics acts as the ultimate search engine for this biological library, and we will also discuss the inherent limitations of these computational tools. To truly appreciate bioinformatics, we must first understand why it matters. As we just discussed, the human genome is a massive string of billions of chemical letters. Finding a specific gene is like finding a needle in a billion haystacks. Before computers, analyzing these sequences was painstakingly slow. Bioinformatics digitizes biology, acting as a super-powered librarian. It allows us to instantly search, compare, and understand these massive biological texts. By bringing computer science into the biology lab, we can run simulations and analyses that would be physically impossible to perform using traditional methods alone. Now that we understand the need for this tool, let us formally define what it is. Bioinformatics is an interdisciplinary field that develops methods and software tools for understanding biological data, especially when the data sets are incredibly large and complex. It is not just biology. It combines computer science, statistics, mathematics, and engineering to analyze and interpret biological information. By digitizing biology, scientists can model complex interactions, predict how molecules will behave, and uncover evolutionary relationships, all within a computer environment before ever stepping foot in a physical laboratory. Building on that definition, let us look at how this is applied in the real world. The applications of bioinformatics are absolutely critical to modern science and medicine. Without it, monumental achievements like the Human Genome Project would have been impossible. It gives us the ability to rapidly sequence and track viruses during global pandemics. In pharmaceutical research, it is heavily used in drug discovery to simulate how a medicine might bind to a disease-causing protein, saving years of expensive trial and error. Furthermore, it is used in agriculture to design crops that are resistant to drought and disease, directly impacting global food security. One of the most powerful applications we just mentioned is searching a database to see if a newly discovered DNA sequence matches anything we already know. But when you search through millions of sequences, you will inevitably find matches just by random chance. How do we know if a match is biologically meaningful? To do this, we use a statistical measure called the expectation value, or e-value. The e-value tells us how many times we would expect to see a match of this quality purely by random chance in a database of a given size. Let us look at the mathematics behind this. This formula allows us to calculate the probability of a random match. The variable e represents the expectation value, which is the number of random hits we expect to see. The variable m represents the length of the query sequence you are researching, and n represents the total length of the database. S is the raw alignment score, measuring how well the two sequences match. K and lambda are statistical constants based on the scoring system. Notice the relationships here. If the database size n increases, the e-value increases, meaning random matches are more likely. But if the alignment score S increases, the e-value drops exponentially, meaning the match is highly significant. Before we move on, let us see this with a concrete example in drug discovery. A researcher finds a new gene sequence of length 100 and searches it against a database of length 1 million. The alignment score is 50, with K as 0.1 and lambda as 0.5. We plug these known values into our e-value equation. Multiplying the constants and lengths gives us a pre-exponential factor of 10 million. The exponent simplifies to negative 25. Calculating e to the power of negative 25 gives a very small number, showing how a high score drastically reduces random probability. Multiplying these gives a final e-value of approximately 0.000138. Because this is much less than 0.01, this is a genuine biological relationship, not a random accident. Now that we have seen the immense power of bioinformatics, we must address its boundaries. Imagine you are playing a highly realistic flight simulator video game. No matter how perfect the graphics are, crashing the virtual plane will never actually hurt you, and flying it successfully does not make you a real pilot. Bioinformatics faces a similar reality check. We can build incredible computer models of cells, proteins, and DNA, but these are still just simulations of reality, not reality itself. A computer might predict that a certain chemical will cure a disease perfectly, but when tested in a real living organism, it might fail completely due to biological factors the computer did not know about. This brings us to the specific limitations of relying on computational methods. One major limitation is the dependence on data quality. If the input data from biological experiments is flawed or noisy, the computational predictions will also be flawed, a concept known as garbage in, garbage out. Another limitation is computational complexity. Predicting how a large protein folds requires so much computing power that even supercomputers struggle. Finally, bioinformatics models often have to make simplifying assumptions, meaning they cannot capture every single interaction happening inside a messy, living cell. Therefore, bioinformatics must always be paired with traditional wet lab experiments to verify computational predictions. One of the most massive limitations we face when dealing with these complex models is the issue of false positives. When analyzing huge data sets, like testing thousands of genes at once to see which cause cancer, we run into a statistical trap. Because we run so many statistical tests simultaneously, random chance guarantees that some genes will look important even when they are not. To manage this limitation and avoid wasting millions of dollars in physical research, bioinformaticians must calculate a specific metric to estimate how many of their computational discoveries are actually fake. This metric is called the false discovery rate, and here is the formula we use to calculate it. The variable FDR stands for the false discovery rate, which is the proportion of our positive results that are actually wrong. The variable FP represents the number of false positives, which are genes the computer incorrectly flagged as important. The variable TP represents the number of true positives, which are the genes that are genuinely important. The denominator represents the total number of positive results the computer gave us. As our datasets get larger, the number of false positives naturally grows, which increases the FDR and forces us to be highly skeptical of our own computational results. Let us apply this formula to a real-world microarray experiment. A bioinformatician analyzes 20,000 genes, and the software flags 500 genes as significant. However, after months of physical lab experiments, The biologist finds that only 400 of these genes are actually linked to the disease. These are our true positives. First, we find the false positives by subtracting the 400 true genes from the 500 total flagged genes, giving us 100 incorrect predictions. We plug these values into our formula, dividing 100 by the total 500 flagged genes. The result is 0.20. This means 20% of the computer's predictions were completely wrong, proving why physical verification is always required. To visualize how we manage these limitations, let us look at this workflow diagram. At the top, we start in silico, using bioinformatics software on a computer to analyze a massive dataset of 20,000 genes. The computer narrows this down to 500 predicted genes. However, as indicated by the red warning triangle, this computational step inherently includes false positives. Therefore, the workflow must move horizontally into the wet lab, represented by the microscope. Through physical in vivo or in vitro experiments, biologists test those 500 predictions to filter out the noise, finally arriving at the 400 true genes. The computer does not replace the lab, it simply tells the lab where to look. We have covered a lot of ground today, so let us summarize the key takeaways. Bioinformatics is an indispensable tool that acts as the computational engine for modern biology, allowing us to process massive datasets and discover drugs. We learned how statistical tools like the e-value help us separate genuine biological discoveries from random noise. However, we must always remember that computational models are simplifications of reality. Metrics like the false discovery rate remind us that bioinformatics is prone to generating false positives. Ultimately, bioinformatics does not replace the traditional biology laboratory. Rather, it acts as a highly intelligent compass that tells biologists exactly where they should focus their physical experiments.